Good morning. I'm continuing on with cell reproduction. I'm going to finish up kind of going over chapter 11, talking about the two steps or the two divisions of meiosis. And then we're going to compare meiosis with mitosis and kind of move on from there to talk about Mendelian inheritance, how different traits or characteristics can be inherited through this process of cell reproduction. Okay, so meiosis, to begin, involves two divisions of a germ cell, a cell that is destined for reproduction. Think of the cell beginning with a nucleus that has two sets of chromosomes, one from each parent. By the end of meiosis 1, that diploid germ cell will be reduced to two haploid germ cells, or gametes. So the chromosome level is reduced by half during meiosis 1. And if you look at prophase 1, you will see some of the characteristics are the same as mitosis. The chromosomes are condensed from chromatin. They become visible. Each chromosome has replicated itself in interphase and has a chromatid. There are two sister chromatids per chromosome. But you may notice crossovers occur. That's the real difference between mitosis and meiosis. And you see that in the figure. You see a blue and purple chromosome. One pair is short and the other is much longer. And you see them fused together in what is called synapsis. The regions where the chromatids overlap and can cross over, those are called chiasmata. And you see a couple of those in particular. You really don't see any chiasmata on the two short chromosomes, but you see a chiasmata on the two long ones. And if you go over to metaphase 1, you will notice that there has been a crossover event. The blue chromosome has a small portion that's lavender or purple, and the purple chromosome has a small portion that's blue. Each of the two small chromosomes did not cross over. You see them as being solid purple and solid blue. So, prophase 1 is one of the unique events in meiosis 1. Crossing over occurs, or at least can occur. The second unique event is in anaphase 1. It does not say chromatids separate. Excuse me. It says homologous chromosomes separate. This reduces the chromosome number in each daughter cell by 1/2. So prophase 1 and anaphase 1 have very unique events that result in a diploid cell dividing to form two haploid cells that are genetically different from one another. That's a very different type of division than in mitosis. We did not talk about cytokinesis. The cytokinesis, the division of cytoplasm, usually is initiated in anaphase 1 and is finished by telophase 1 to form two daughter cells. You might notice the bullet in metaphase 1 is pretty much the same as for mitosis, and the bullet in telophase 1, those characteristics are the same as well. So the difference that really arises in meiosis, the differences are in prophase 1 and anaphase 1, crossing over and separation of homologous chromosomes. Those are your two unique events that occur. Meiosis 2, you start out with two cells that are haploid. Each cell divides and forms two cells. So at the end of meiosis 2, you have 4 haploid cells. This division is often referred to as a mitotic division because there is no crossing over. When you start in prophase 2, each chromosome is already duplicated. And in anaphase 2, the chromatids, the sister chromatids, will separate. So you start out with cells that are haploid, and you end with cells that are haploid. So there is no crossing over that occurs, and there's no reduction in the chromosome number. So this is basically a mitotic division, just like mitosis. All of the bullets under each phase are basically the same as what you would see in chapter 10 for mitosis. So in prophase 2, the chromosomes become visible, the spindle forms, and the membrane around the nucleus disintegrates. In metaphase 2, the chromosomes will migrate toward the equator, the equatorial plane or center of the dividing cells. In anaphase 2, the spindle fibers contract and the chromatids separate and move to the opposing poles of those two cells. Cytokinesis is probably initiated, and it completes in telophase 2 to divide the initial 2 cells into 4. In telophase 2, the chromosomes arrive at the poles of the dividing cells. A nuclear membrane forms around them, and the chromosomes decondense and lengthen into what is called chromatin. In each cell, we started with 4 chromosomes in meiosis 1 in this example, 2 blue and 2 purple. And at the end of meiosis 1, we had 1 purple and 1 blue. We had 2 chromosomes. We had 2 haploid cells. In meiosis 2, we started with 2 haploid cells with 2 chromosomes, and they end up, after the chromatids separate, with 2 chromosomes in each of those 4 cells. So the chromosome number has been reduced by half, and the possibility exists for each haploid cell to be genetically distinct or genetically different from the other 3. Again, note, if these haploid cells are male, all 4 of them typically survive. If these 4 haploid cells are female, there only tends to be 1 that survives. The other 3 disintegrate. So you have 2 different types of cell reproduction in eukaryotes. Mitosis for repair and growth and maintenance of cell number. There are some eukaryotes that can use mitosis to reproduce. Plants are a real good example of that. Meiosis, you have a germ cell that divides twice to form 4 daughter cells. The daughter cells may be genetically distinct from each other and from the parent. The 2 unique events in meiosis are crossing over in prophase 1 and the separation of homologous chromosomes in anaphase 1. So mitosis and meiosis are very important types of cell reproduction events in eukaryotic cells. The last 2 slides, I just wanted to point out to you that when sperm are formed in animals, you start with a diploid cell. You end up with 4 haploid cells, and all 4 of those would typically mature as sperm. The formation of ova in particular in the female human, often begins when she is a fetus inside of her mom. And the formation of ova begins and stops usually in prophase 1. And by the time a female is born, most all of the eggs or ova that she's going to produce in her life have already been formed. I believe it's been shown that some females can produce eggs after they're born, and once they reach puberty, there are eggs that can be formed, but it's a small percentage of the total. So sperm cells are produced throughout the life of the male. Eggs are typically produced within a 20 to 30 year period. Of the 4 products in a female from meiosis, only 1 tends to become an ovum or egg, and the other 3 degenerate. They're referred to as polar bodies. You can see in the picture the difference in size between an egg and a sperm, just to note the difference in size there. So that kind of ends the topic of meiosis. There's some good material for you to read in chapter 11 in the online OpenStax textbook, and there's lots of practice questions for you to do as well to kind of pace yourself through this material and kind of familiarize yourself with mitosis and meiosis as cell reproduction events, and then how they might be used in inheritance of characteristics, which we refer to as genetics. So our next topic is going to be genetics, in particular those as described by Gregor Mendel. So y'all take care, and talk to you soon. Bye.